This conference will now be recorded.
So, good evening to one and all. So, for the last 2 days, we were dealing with the topic fermentation. We were going through basics of fermentation regarding fermenter design and all other aids which is used for a successful fermentation process in order to obtain the product, desired product in the maximum concentration. And then we have started the second topic, that is production of micro and macromolecules by fermentation, and we started with pharmaceuticals. So already I hope you remember till where we discussed last class. So pharmaceuticals already we have gone through this. It include antibiotics, steroids, vitamins, vaccines, etc. Fine. Yeah. Yes, Vidushi. And we started with antibiotics. The first antibiotic we completed was penicillin, regarding what are all the characteristic features of penicillin production, fermentation, Anaka. Yes, I hope all of you remember. Okay, fine, good, thank you. So now we are continuing with the pharmaceuticals, that is the antibiotics. We are continuing with the fermentation of antibiotics. Now we are going to study about another one or two classes of antibiotics. First one is cephalosporins, and the second one is streptomycin. So you have heard about all these antibiotics, penicillin, streptomycin, tetracycline. You know what are antibiotics. Antibiotics are nothing but biochemical substances produced by microorganisms, certain class or certain group of microorganisms which can inhibit or kill other closely other related species or different species. For example, antibiotics produced by fungus can inhibit or kill the bacteria. Fine. So actually there is a story behind the invention of penicillin by Alexander Fleming. Do anyone know about that story? Can you type in the comment box? Do you know about the story? How penicillin was discovered by Alexander Fleming? Yes, you might have read about that story, right? Regarding the penicillin production. I think, yeah, Vidushi and Anaka knows about the story. Any other person? Okay, so you might have heard about that story. I don't want to repeat it. It's a very simple story. Like Alexander Fleming, he was working on bacteria, a particular group of gram-negative bacteria or something like that. So during his vacation, he keep the bacterial plates near the fungal culture, and he went for vacation. So once he came back, he has seen that the fungal spores has somehow fall on the bacterial plate, and on that area where the fungus has grown, mycelia has grown, the bacteria has not grown. It has inhibited. He has observed the first inhibition zone of an antibiotic. Then he started study about the biochemical substance which was responsible for that bacterial inhibition, and finally he discovered penicillin. So this was the history behind invention of penicillin by Alexander Fleming. Just I mentioned about it. Okay, so we will start with cephalosporin. Now cephalosporins are another class of antibiotics. For example, so while we were talking about penicillin, we have already mentioned the common form of penicillin which is found is penicillin G, which can be transferred into other forms by certain substitutions as well. So here also the common type of cephalosporin which is fermented is cephalosporin C, like penicillin G. It is made as a fermentation product of another fungus, that is Cephalosporium acremonium. Now this form is not potent enough for clinical use. So the cephalosporin produced as a fermentation product of this particular fungus, that is Cephalosporium acremonium, in its crude form it is not active, or it is not potent for a clinical use. It will not act as an active biochemical component. So what we have to do, we have to modify it chemically. Now this product, this fermentation product of Cephalosporium acremonium, nothing but Cephalosporin C, is modified chemically to form clinically useful products with broad spectrum of antibacterial action. Broad spectrum of antibacterial action. Can anybody tell me what do you mean by broad spectrum of antibacterial action? Anybody can type in the comment box. What do you mean by broad spectrum antibiotics and narrow spectrum antibiotics? Anybody has any idea about that? Anaka, Vidushi, then all are there. Maunia, yes. Today we have Anaka, Shalini, Maunia, Vidushi. 4 of you. Anaka, yes, Anaka, Shalini, Maunia, Vidushi. Who is absent today? Prema Kumari is absent today. That's it. Only one person absent today. I think 6 people were there together, right? Yes. So anybody? I hope you all are listening to the class. Do anybody have any idea about broad spectrum antibiotic and narrow spectrum antibiotic? If you have, just type in the comment box. Otherwise, you tell me no or sorry, sir. That's it. No. Okay, Anaka don't know. Any other person? Shalini also don't know. Then Vidushi. What about Vidushi? And yes, Vidushi. Broad spectrum works against wide range of bacteria. Excellent. That's it. That's it. Very good. So that's it. Fine. So 2 classes. Antibiotic can be broadly classified into 2. We can tell some of the antibiotics are broad spectrum antibiotics. Some of the antibiotics are narrow spectrum antibiotics. Broad spectrum antibiotics can act upon a wide range of bacteria, maybe gram positive, gram negative, different types of bacteria. This antibiotics is active against. Whereas narrow spectrum antibiotics can be applied against a particular class of bacteria only. Do anyone has done the practical antibiotic sensitivity test in your classes, graduation or post-graduation classes? Did anyone do antibiotic sensitivity testing? Yes, Anaka has done. Then Vidushi has done. What about Maunia and Maunia has also done. So you know actually. Then once more, Vidushi, Vidushi, Anaka, Maunia. Then Shalini. What about Shalini? Yeah. So all of you know antibiotic sensitivity testing. So that's why we are performing antibiotic. So before applying a dosage of antibiotic for a particular disease, doctors will go for antibiotic sensitivity testing because they want to know which antibiotic is resistant against that particular class of bacteria which can actively inhibit them or kill them. Then only he will select the appropriate antibiotic. So similarly, we were talking about the broad spectrum antibiotics. Example for broad spectrum antibiotics, penicillin, cephalosporin, lot of antibiotics are there. Fine. Now, so here cephalosporin is also a broad spectrum antibiotic, but once we get the fermentation product as cephalosporin C, it is not clinically potent or clinically efficient to be applied. We have to go for some chemical modification so that it will form a potent broad spectrum antibiotic. Fine. For example, 3rd generation cephalosporins like moxalactam. See, moxalactam is a derivative of cephalosporin, a chemically modified cephalosporin antibiotic. Example is moxalactam. It is a 3rd generation antibiotic. Okay, after 1 or 2 chemical substitutions, we have got this moxalactam. Now, it can resist bacteria that produce enzymes capable of degrading penicillin and cephalosporin. Okay. Have you heard about the term antibiotic resistance bacteria? Antibiotic resistant bacteria. Have you heard about the term? Okay. Vidushi has heard about antibiotic resistance bacteria. Anaka has heard. Shalini has heard. Maunia. What about Maunia? Have you heard about antibiotic? Very good. So antibiotic, from the term itself, you know what do you mean by antibiotic resistance bacteria? They are bacteria which can produce enzymes which are capable of degrading antibiotics so that it can even grow in the presence of antibiotics. So those organisms or those bacteria which can produce such enzymes, they will be resistant to the antibiotic, so that those organisms which cannot degrade the antibiotic, they will be sensitive. So when doctors, before preferring a antibiotic, what they will do, they will go for antibiotic sensitivity testing and they will take the maximum active antibiotic which can kill or which can inhibit wide range of bacteria. Fine. So antibiotic resistant bacteria are those bacteria which can produce enzymes which can degrade antibiotics like penicillin and cephalosporin. These bacteria can produce enzymes which can degrade this antibiotic. Fine. Can you give me an example for a antibiotic resistance bacteria? Anybody in the comment box? Example for an antibiotic resistance bacteria. There is a very famous class of organisms, gram-positive. Lot of studies are going on in this antibiotic resistant group. Anybody? Any example for antibiotic resistance bacteria? Maunia, Shalini, Vidushi, Anaka, anyone? Can you give me an example? Anybody? Have you heard the term MRSA? Anybody has heard the term MRSA? Anybody knows the full form of MRSA? Anyone? Maunia? No. Okay. Maunia don't know. What about Shalini, Vidushi and Anaka? Okay. MRSA is nothing but methicillin resistant Staphylococcus aureus. This is MRSA. Very good, Vidushi. Methicillin resistant Staphylococcus aureus. They are a group or a class of Staph aureus species which is highly resistant against the antibiotic methicillin. So it is causing a lot of pyogenic infections, pus forming infections in such patients. It's a very famous antibiotic resistant bacteria. Okay, aureus have only one A. Okay, I'm sorry. Maybe this is aureus, not like this. Okay, the spelling of Vidushi is correct. Methicillin resistant Staphylococcus aureus. Very good. Fine. So it's an example for an antibiotic resistance bacteria. So here the third generation of cephalosporin can produce. That is, for example, moxalactam has got the ability to act even against antibiotic resistance bacteria, which has got the power to degrade penicillin and cephalosporin. Clear about cephalosporins? And the next category is streptomycin. So streptomycin is also like penicillin, one important antibiotic resistant, one important antibiotic. Okay, now they are produced by strains of actinomycetes. What are actinomycetes? What are actinomycetes? Have you heard the term actinomycetes? Anybody? It's a type of fungus. Is it actinomycetes a fungus? Who told you? Vidushi. Then what is a fungus? Vidushi, if actinomycetes is fungus, then what is fungus? Okay. Now what about bacteria? Vidushi. Okay. If actinomycetes is bacteria, then what is bacteria? Okay, Vidushi and Anaka, tell me. You are telling actinomycetes is bacteria. Then what is a bacteria? What about the other two people? Any comments on actinomycetes? Any particular characteristic of actinomycetes have you studied? Yes. So Anaka has told it is bacteria. Vidushi has told it is bacteria. Then Vidushi has told in the beginning it is fungus. Phylum of G plus bacteria. Okay. Fine. See, it is actually actinomycetes are a class of bacteria only. Fine. Gram positive bacteria. G plus C. Yeah, it's correct actually. You have written. Fine. So the thing is, it is have you there is a, see, when rain comes after a long time, an earthy odor will come, you know. Have you experienced it? I am not sure. During summer season or during summer rain, when the first drop of rain falls on the soil, you will get an earthy odor. It is due to the activity of actinomycetes actually. Fine. They are a class of bacteria only. Fine. Okay. So just when we read about actinomycetes, I just tell in the middle. That's it. Fine. So streptomycin is produced by a strain of actinomycetes. Now the organism is Streptomyces graecosus. Fine. Now it also produces numerous other antimicrobics which inhibit protein synthesis in prokaryotes. So that is the speciality of antibacterial substances produced by this Streptomyces graecosus. It can inhibit protein synthesis. So antibiotics acts on bacteria in many ways. Some of the antibiotic they will cleave the cell wall. Okay. Some of them will inhibit the protein synthesis. Some of them will inhibit a replication of bacteria. So actinomycin, each antibiotic has got a mode of action. Here The antibiotics produced by the actinomycetes, Streptomyces graeus, that is streptomycin and many other antibiotics, which has got the ability to inhibit protein synthesis in bacteria. So that is the mode of action of streptomycin. Now, this antibiotic was discovered by scientist Selman Waxman. Fine. Now, how we are going for the fermentation? First of all, similarly like our penicillin, here also spores. First of all, we are going for scaling up of the inoculum. Spores are first inoculated into a medium to produce a culture with high mycelial biomass or fungal biomass. That's why mycelia. It's actually we call actinomycetes as filamentous bacteria. Sometimes we call it as filamentous bacteria. It resembles fungus in certain character and it resembles bacteria in certain character. Usually we call it as bacteria. We classify it as actinobacteria, but still it has got some ability. It has got some characteristic features of fungus. Filamentous bacteria we call actinomycetes. Fine. So here that's why we call it as mycelial biomass. Mycelium is nothing but the fungal body. Fine. So here first of all, cells are spores are inoculated into a medium to produce a mass or bacterial actinomycetes culture. Then we are converting it into an inoculation tank in order to produce more amount of inoculum. Then we are going for a production tank. See how we are going for scaling up of inoculum. We started with maybe a conical flask, then we went to a small inoculation tank with maybe a, like as I have explained in the last class, maybe 4 liter to 40 liter to 400 liter to maybe 4000 liter, like that. Same way. Okay. So in the production tank, we are going for our large scale fermentation process. Fine. Okay. So we are continuing. Now, what about the other characteristics of fermentation of streptomycin? Now, the basic medium consists of what all things? Soybean meal as nitrogen source. So, as I have already explained, a medium must contain nitrogen source, carbon source, phosphorus source. All those things must be there. So here, soybean meal is the nitrogen source, glucose is the carbon source, and then sodium chloride. Fine. Now, optimum temperature is around 28 degrees Celsius. Maximum production rate is achieved in the pH is about 7.6 to 8. Now, high rates of aeration and agitation is required. So, the process, the sparger and baffles must be function properly and continuously in order to provide high amount of aeration and agitation mixing. Fine. Now, process lasts for approximately 10 days of fermentation, long time. Fine. Now, yield is approximately maybe more than 1 gram per liter of the fermentation media. Now, what is happening after that? So, streptomycin accumulation happens in the second phase as secondary metabolite. So, you know what is secondary metabolite and primary metabolite. Already I have explained it. Primary metabolites are those substances which is directly required by the organism for its growth and survival, such as for replication, protein synthesis, cell formation, all those things. Secondary metabolites are those substances which are not directly needed by the organism, but it is much needed by people, it is much needed by humans for commercial production. For example, most of the antibiotics, hormones, enzymes, all are secondary metabolites. Fine. Now, after the process, mycelium is separated from broth by filtration. So we will filter it to separate the biomass, actinomycetes biomass. Then streptomycin is recovered. Now it is a water-soluble substance. Fine. It's a water-soluble basic substance. Fine. So that it is insoluble in most organic solvent. It is soluble in water but insoluble in organic solvent. So how we are going for recovery? By absorbing on activated charcoal. We will absorb streptomycin on activated charcoal, and then after activation, in the next step, we will elute it out by acid alcohol. Fine. After that, the antibiotic is precipitated with acetone and purified using column chromatography. So we are going for, after the fermentation, after the formation of streptomycin, first of all, we will separate it from the bacterial or the mycelial biomass by means of filtration. Then we will allow it to adsorb onto an activated charcoal. After adsorption, we will elute it out using acid alcohol. Then we will precipitate it with acetone and finally purify it with column chromatography. So these are all the various steps involved in the purification of streptomycin. Clear? Production of streptomycin. Any doubts, please do ask. Fine. So this is all about the production of streptomycin. Yes. Yeah. Thank you. This is about the production of streptomycin from the master culture. You can see here one by one. See, we started with the master culture. Fine. Then we are going for shaker flask. Then we are going for seed vessel. Then we are going for fermenter. Step by step. Fine. Then see filtration, neutralization, clarification, vacuum oven. A lot of functions are there. Finally, this is about the whole chart on the industrial production of streptomycin. How we are going for industrial production of streptomycin right from the starter culture up to the dispatch, packing and dispatch, just for your information. So this is a chart which I have obtained from one textbook. You have got a lot of antibiotics with the organisms, cultures. This is most important for your multiple choice question. See, lot of antibiotics. Right from the beginning, you can see amphotericin B, bacitracin, carbamazepine. So just go through this. For example, the antibiotic chloramphenicol. What is the industrial organism? Streptomyces venezuela. Fine. For erythromycin, it is Streptomyces erythraeus. For penicillin, Penicillium chrysogenum. Then for tetracycline, it is dechlorination and hydrogenation of chlor tetracycline, direct fermentation in dechlorinated medium. So just go through this chart. Fine. So you can crack any multiple choice question which will come regarding antibiotics. Name of the antibiotic and causative organism and producing organism, industrially important organism for the production of antibiotic. Clear? And now we are going to the next class of pharmaceuticals, steroids. So that's all about antibiotics. Now we will start with steroids. It's the second class of pharmaceuticals, steroids. So first of all, what are steroids? You have heard about the term steroids a lot. So steroid hormones can regulate various aspects of metabolism in animals. It will control metabolic activities in animals, including humans. Metabolism means living activities, anabolism plus catabolism. Now, for example for a steroid is cortisone. Cortisone is found to relieve pain associated with rheumatoid arthritis. So we can use the steroid cortisone for the treatment of rheumatoid arthritis. Fine. Now, various cortisone derivatives are also useful with allergic and other undesired inflammatory responses of human body. So we can use cortisone for the treatment of rheumatoid arthritis. Also, some of other derivatives of cortisones are effective for treatment of allergic and other inflammatory body issues. Fine. Now, various steroid hormones regulate human sexuality. That is, some of the steroid hormones will also regulate human sexuality. Fine. Then, property of steroid depends upon the nature and exact position of chemical constituents on the steroid ring structure. Okay. So, steroid has got some ring structures, so that you will have some chemical constituents on the ring structure. So, property or the how a steroid will act on human body, it depends upon the nature and the position of the steroid ring structure. Nature of the chemical. For example, if hydrogen is present in second position, or maybe hydroxyl group is present in fourth position of the steroid ring. So based on that, we can get some idea, or we can generate idea about the nature and property of steroids. Fine. Now, chemical synthesis of steroids is very complex, very difficult. That's why we are going for microbial synthesis. For example, cortisone synthesized chemically from deoxycholic acid process requires 37 steps, very tedious activity, and under extreme conditions of temperature and pressure with huge production cost. So steroids, the chemical synthesis of steroid is highly complex and cost involving than microbial synthesis of steroids. For example, the chemical synthesis of cortisone from deoxycholic acid requires 37 steps. Then extreme environmental conditions such as temperature and pressure, and also huge production cost. Now, That is, the major challenge is to introduce an oxygen atom at number 11 position of the steroid ring. So that is, so when we are going for microbial synthesis or microbial fermentation production of steroids, we will be going for some chemical substitution using microorganisms. Microorganisms can perform any chemical substitution, unlike chemical action. You have to grow some you have to grow microbial culture, the required microbial culture. It can do hydrogenation for you. What do you mean by hydrogenation? Addition of hydrogen. It can do dehydrogenation for you. What do you mean by dehydrogenation? Removal of hydrogen. Fine. Hydroxylation, addition of hydroxyl group, OH group. So for this chemical substitution, we are using microorganisms or bacteria during the synthesis of steroids. Clear? Now, for example, now two organisms you can see, Rhizopus arizus and Cunninghamella blaxliana. Very difficult to pronounce, and be careful about the spelling as well. Rhizopus and Cunninghamella. Rhizopus arizus and Cunninghamella blaxliana. So they are two fungal strains used for steroid production or chemical substitution for the production of steroids. For example, microbial transformation such as hydrogenation, addition of hydrogen, dehydrogenation, removal of hydrogen from the ring, then removal and addition of side chain. All those activities can be performed by bacteria, and it is less cost with better product recovery. Fine. For example, the microorganism that is Rhizopus arizus, how we are doing it? It is grown in a fermenter with an appropriate growth medium, incubation, aeration, agitation, everything. We are growing it in a fermenter. Then the steroid to be transferred is added to this medium. For example, if progesterone is added, the steroid is hydroxylated at position 11. So for example, if we have to perform a hydroxylation for the steroid progesterone, what we will do? We will cultivate Rhizopus arizus in a fermenter on an appropriate growth medium with proper incubation conditions, that is temperature, pH, humidity, proper aeration, agitation, everything, so that we will get a bacterial biomass, I mean fungal biomass first of all. Then we will add the steroid. For example, if it is progesterone, we will add progesterone into the medium, so that due by bacterial action, it will add a hydroxyl group or OH group at the position 11 of this steroid progesterone. And finally, the product is recovered by extraction using methylene chloride or other solvent. Solvent extraction will be performed. Now, chromatographic purification, purified chromatographically and recovered by crystallization. Once chemical substitution has taken place by the bacteria, what we will do? We will recover the product, the steroid, by solvent extraction using methylene chloride, followed by purification using chromatography, followed by recovery using crystallization. That's all about the production of steroids. Is that clear? Is it clear till this point what is happening? Yes, everybody, is it clear? Yes, Vidushi, yes, Anaka, Maunia, very good, good response, thank you. And we are moving on to production of human proteins, third category of our pharmaceuticals, human proteins. Now, so here comes our rDNA technology, recombinant DNA technology. Using recombinant DNA technology, human DNA, which code for various proteins, is incorporated into genome of bacteria. So we are using rDNA technology for production of human proteins. What we will do? We will identify the area of human DNA which is responsible for the production of a protein. Then, using a proper cloning vector such as plasmid, using our restriction enzymes, fine, we will restrict that portion. Then we will ligate it or fix it with our plasmid, and then this plasmid will be sent into a bacteria for multiplication and expression of that particular gene, so that you can get the particular protein. Fine. So now these recombinant bacteria are grown in fermenters for commercial production of human protein. Now we can, once the successful recombinant DNA technology has taken place and we have got recombinant bacteria with human protein coding genes on plasmid, we can go for a fermentation of this bacteria. We can produce it in large quantity for commercial production of these proteins. Now, what are all the proteins? For example, human insulin is produced by recombinant E. coli strain, human insulin, and marketed as humulin. So, the protein human insulin is naturally produced in human body. Now, we can produce it by recombinant DNA technology using recombinant E. coli strain, and we market it as humulin. That is the marketing name, brand name. Fine. Now, other strains used to produce HGH. What is HGH? Human growth hormone, TNF, tumor necrosis factor, interferon, then interleukin 2. So, these are all the examples for human proteins produced by bacteria. What are they? Examples are human insulin, HGH, human growth hormone, TNF, tumor necrosis factor, interferon, and interleukin 2. Now, what is the action of each and every one? Humulin is used to treat diabetes. Fine. HGH, human growth hormone, is used to treat dwarfism. Okay, dwarfism and gigantism, two extreme conditions of human body. Dwarfism means less growth, gigantism means high level of growth, uncontrolled growth. Fine. So here, humulin or human insulin produced by recombinant E. coli strain is used to treat diabetes. HGH, human growth hormone, is used to treat dwarfism. Then TNF, interferon, and interleukin 2. They are components of natural human immune response. So it is used for proper immune response and in order to protect human body from dreadful bacteria and viruses or diseases. Now, interferon is important in defense against viruses. It is an antiviral human protein. Example is interferon. Now, TNF, that is tumor necrosis factor, produced by macrophages. They are a group of cells, kill cancer cells. You have heard about macrophages. Macrophages are a group of immune cells. Okay, so tumor necrosis factor produced by macrophages will kill cancer cells and other infectious microorganisms. Fine. Now, synthetic peptides are also developed, including blood clotting factors, then tissue healing factors, growth factors, etc. So, by means of recombinant DNA technology, it is possible to produce recombinant human proteins using bacterial strains such as E. coli with added advantage, added functions, or high efficiency. Fine. Examples are human insulin, humulin, HGH, human growth hormone, TNF, tumor necrosis factor, interferon, interleukin 2, then our synthetic peptides. All these are examples for our human proteins. Clear? What are human proteins? What are the activity of each human protein? What is the action of each human protein? How we are going for the formation of human protein? Yes. Yes, Vidushi, thank you. So here again comes our chart for your reference. I have added as far as maximum information for you to refer and learn. It's a self-help study. Yes, Anata, thank you. Insulin, for example, product name is humulin or novolin. That is the brand name. Now, what is that? Hormone which regulates sugar level in blood, used in the treatment of diabetes. Like that, this chart contains details about insulin, human growth hormone, bone growth factor, interferon alpha, beta, gamma, interleukin 2, TNF, that is tumor necrosis factor, tissue plasminogen activator, blood clotting factor 8, epidermal growth factor, granulocyte colony stimulating factor, and erythropoietin, with their brand names and their functions and use. Please go through all these charts. Okay, you can expect multiple choice questions from these. Fine, good. Thank you, Vidushi. We are moving on. Yeah, thank you all, Anaka, Shalini, Vidushi, and yeah. So finally, we are moving on to, now we have vaccines. Now what all things are left? Today we will finish this topic, pharmaceutical. We have vaccines to learn, then we have vitamins. That's it. Vaccines and vitamins remain. We will finish it. So next we are going for vaccines. So vaccines is most important nowadays. Our corona vaccine is in the news now. So what do you mean by a vaccine, first of all? Can anybody give me an idea about vaccine? Define a vaccine. Shalini, Anatha, Maunia, Vidushi. Question is for you, 4 of you. What do you mean by a vaccine? What is your idea about vaccine? Or what is the difference between a vaccine and an antibiotic? Tell me. Anybody? Anybody in the comment box? Anybody can come to the comment box and answer. Or even you can unmute your mic and speak. If you want to tell long sentences, you can unmute your mic and speak. No problem. You are welcome. What are vaccines? Yeah, Shalini. A killed microorganism which is used to stimulate the immune response. Okay, I do agree partially. Kill the microorganism. Okay, then please explain. If we kill a microorganism, for example, a bacteria or a virus, then how can it stimulate the immune response? If we are killing it, killing means it is die, then how can it stimulate our immune response? Or how can it stimulate our immune system? When we inject a killed microorganism as vaccine into a human body, it must activate. The microorganism will act as an antigen. Okay. So we kill the microorganism, still it acts as an antigen. Fine. Okay. Good. Good answer. Then anybody else? See, vaccines can be preparations of, as Shalini has told, killed or live attenuated microorganisms. What do you mean by attenuation? Attenuation means inactivation. Fine. Either the organism will be live but attenuated or inactivated, or it will be in the killed form. But in both form, the organism, this injection or the preparation of a vaccine has less antigenicity but more immunogenicity. What do you mean by that? Less antigenicity and more immunogenicity means, of course, it imitates as an antigen in the human body. When we inject a vaccine into the body, this vaccine will imitate as an antigen, but it will not cause any disease, but it will lead to the formation of antibodies against the antigen, so that a second challenge with the same antigen will stimulate our immune system in a faster pace, thereby providing antibody mediated immunity and cell mediated immunity. Have you heard about B lymphocytes, B cells, antibiotic producing, antibody producing, then T lymphocytes, T cells, cell mediated immunity? You might have studied in immunology. Fine. So vaccines are preparations of microorganism, bacteria, virus, whatever it is, which must be live, attenuated, live and inactivated or killed one. But it has less antigenicity but more immunogenicity, which means it won't cause any disease, but it will lead to the formation of antibodies against that particular antigen, so that a second challenge with the real pathogenic organism will stimulate our immune system in a faster pace, which will provide a better protection. Clear? That are nothing but vaccines. So they are used to prevent. No. One more question is there remaining. My second question nobody answered. What is the difference between a vaccine and an antibiotic? That is my second question. What is the difference between a vaccine and an antibiotic? Anaka, Shalini, Maunia. I think Maunia and Vidushi has slept. They slept because the class is a dry class, boring topic, so I think they slept. What about Vidushi and Maunia? Are you there or have you slept? If you are there, tell me the difference between an antibiotic and a vaccine. Yes, Maunia. Vaccine is to produce antibody. Okay, I do agree. But then what about antibiotic? And what about Vidushi? Are you there, Vidushi? Substance produced by microorganism is known as antibiotic. Okay, then what about vaccine? Vaccine is the bacteria itself. Okay, let me, I agree to that partially. See, the most important is in the mode of action. Most important thing you have to tell is mode of action. Yes, there you are, Maunia. Antibiotics is given to cure disease. Then what about vaccine? What about vaccine? Tell me, Maunia. You are near the answer. Antibiotic is given, vaccines are to prevent. Excellent, Vidushi. Excellent. That's it. Fine. See, vaccines are proactive, but antibiotics are reactive. Reactive and proactive. It is very big difference between antibiotic and vaccine. Fine. Vaccines are used to prevent diseases, where antibiotics are applied to cure diseases. There is antibiotic therapy where antibiotics are using as vaccines before the disease. There is a section like that, but you leave it. Generally, when we are speaking, antibiotics will cure diseases, vaccines will prevent diseases. Then, as she has told, antibiotic is produced by certain organisms. They are biochemical substances secreted by certain organisms. But vaccines are the organism itself. It can be either the whole organism or a part of the organism, which can stimulate the immune system of the recipient to generate antibody. Fine? Good. So, now we are going to our topic very fast. Vaccines are used to prevent various diseases, involve growing microorganisms possessing antigenic properties needed to generate a primary immune response, produced by mutant strains of pathogens or by attenuating either mutant strains of pathogen or attenuating or inactivating virulent pathogens without removing antigens necessary to generate immune response. Fine. Viral strains are grown in embryonated eggs, also produced by tissue culture to prevent any allergic responses. Commercial vaccines are tested and standardized before use. Now we are seeing the various phases of our vaccine production. Corona vaccine, COVID vaccine, phase 1 trial, phase 2 trial, phase 3 trial, human trial. Lot of things are there. Fine. Excuse me. Okay, we are back. So, so that's about vaccines. Fine. Now we are going to our next topic. Okay. Okay. So these are again a chart for your reference. Disease, which disease, which has a type of vaccine. So you can see there are some terms. I can explain the terms for you. See here. You can see attenuated live virus. See, attenuated live virus, which means the virus is live but inactivated. It is inactivated. Attenuation means inactivation. How we are inactivating a live virus? Do you have any idea about the vaccine production? How we will inactivate a virus? A virulent, yeah, it can be a virulent, but it has got the similar antigenic structure. Yes. See, for example, there is a route of entry for any virus. For example, our coronavirus, the route of entry is by respiratory system. So a virus which is entering through the respiratory system, if we inject that virus through the digestive system, what is going to happen? What is going to happen? Just think. It may lead to sometimes it may not be able to go for a successful successful disease. Fine. It's a type of attenuation. Then you can use some chemicals like formaldehyde, even UV. Many things are there for inactivation of viruses. Fine. So here all, see, measles, mumps, rubella, attenuated live virus is used. Then you can see here polio inactivated virus. Fine. So in both cases, only one thing is happening, whether it is attenuation or inactivation. What we are doing is we are making the virus not able to cause a successful disease, but it is able to generate the antibody production in the recipient. That's what the simple example, simple meaning. You can see inactivated bacteria here in pertussis, cholera. Then toxoid, there's a term, toxoid. What do you mean by toxoid? Anybody? What is toxoid? Anybody? Toxoid. It's nothing but inactivated toxin. Toxin can also, see, everything is antigen, whether it is a toxin, whether it is a live organism, whether it is a killed organism. The cell wall of the organism may contain some antigenic structures. Toxoid is nothing but it is toxin, but inactivated toxin. Inactivated toxin, that is toxoid. Fine. Diphtheria toxoid, tetanus toxoid. Fine. So similarly, it can generate antibodies in the recipient, but it will not be able to cause a disease. Fine. Then for there we are also using some treated capsular material as vaccines. Clear? Vaccines? Any doubt in this topic? Vaccine? Anybody? Yes, Shalini, Anaka, Maunia. Then is clear? Vaccine? Yeah, good. Anaka, Shalini, Maunia, and Vidushi. Okay, fine. Now we are going to our last topic today. Then we will have a few multiple choice questions to do. Yeah, production of vitamins. Yeah, last topic. Fine. So what are vitamins again? So now we have studied about antibiotics. We have studied about vaccines. We have studied about human proteins. Fine. Now all those things will come in the category pharmaceutical. Now we are going for vitamins. Vitamins are essential nutritional factors. Okay. Produced by microbial fermentation and used as dietary supplements. Dietary supplement means together with our diet, like our food actually. Fine. For example, vitamin B12 produced as a byproduct of Streptomyces antibiotic fermentation. Fine. Now for that we are adding a soluble cobalt salt as precursor. Precursor is nothing but the starting material for the production of vitamin B12 during Streptomyces antibiotic fermentation. We are adding cobalt salt as a starting element, starting component, that is precursor. Fine. Now vitamin B12 is also produced commercially by direct fermentation using Propionibacterium shermani or Paracoccus denitrificans. Now what we are doing in first case, Propionibacterium shermani grown in an anaerobic culture for 3 days, then in aerobic culture for 4 days to produce vitamin B12. Okay. Now growth medium contains glucose, corn, steep liquor, cobalt, chloride. All these things are components which will act as carbon source, nitrogen source, phosphorus source. The medium maintained at pH 7 using ammonium hydroxide. Then Propioni Paracoccus denitrificans are grown for 2 days in aerated culture for vitamin B12 production using a medium containing sucrose, glutamic acid, cobalt chloride, 5,6-dimethyl benzenamidazole and salt. So these are all the media composition, the pH condition, then the aeration condition. Fine, all those things required for the production of vitamin B12 using 2 different bacterial strains. Okay. Yes. So now riboflavin. Riboflavin is another vitamin. Riboflavin is produced as a fermentation product by various microorganisms. Byproduct of acetone butanol fermentation produced by various Clostridia species. Okay. Commercial production of riboflavin is by direct fermentation using fungal species Erythrothecium ashby or Ashbia gossypi. So this vitamin B12 or riboflavin, all those things are either produced as byproduct of certain fermentation or as by direct fermentation. In both ways we can produce it. For example, riboflavin can be produced as a byproduct of acetone butanol fermentation using Clostridia or by direct fermentation using Erythrothecium ashby or Ashbia gossypi. Now, the medium contains glucose and corn oil. Fermentation using Ashbia gossypi is normally carried out at 26 to 28 degrees Celsius. pH is 6.7 to 6 to 7.5, approximately 4 to 5 days. After fermentation, cells are recovered and used as feed supplement to supply needed riboflavin for animals. Okay, so this is about the production of vitamins, vitamin B12, riboflavin. What are all the various conditions as byproduct by direct fermentation? All these things are possible. Clear? Here also you have got a chart for your reference. Riboflavin, what is the name of the vitamin, culture, then media composition, fermentation condition, and yield. You have riboflavin, you have L-sorbose, you have 5-ketogluconic acid, and vitamin B12. So that's all about our production of pharmaceuticals. So is that clear, this topic till here, everybody? All our 4 participants today, is it clear? Anaka, Vidushi, Maunia, Shalini. By this our first set of fermentations, that is fermentation of pharmaceuticals, comes to an end. Before winding up the class, we will go for few MCQs. As usual, I am going to ask one by one. You have to give me the answer. Okay. First question goes to Maunia. Dash is a protein synthesis inhibitor antibiotic. Option A, cephalosporin. Option B, penicillin. Option C, streptomycin. Option D, tetracycline. Once again, question and options. Dash is a protein synthesis inhibitor antibiotic. Cephalosporin, penicillin, streptomycin, tetracycline. Maunia is going with option A, cephalosporin. What about the others? Shalini, Anaka, Vidushi is going with C, streptomycin. What about Anaka and Shalini? Maunia has given option A. Shalini has given option C. What about Anaka? Everyone is going with option C. Only Maunia going with option A. Maunia, the answer is streptomycin. We have told this point during that. Streptomycin produced by Streptomyces, the actinomycetes. Fine. Streptomycin and many other antimicrobics, their main action is inhibition of protein synthesis. With this, it's okay. The second point, the second question goes to Shalini S. Chemical synthesis of steroids are comparatively less complex than microbial transformation. A, true. B, false. C, hypothetical statement. D, insufficient data. And the question goes to Shalini S. Chemical synthesis of steroids are comparatively less complex than microbial transformation. What about Shalini? What is your option? A, B, C, or D? Shalini is telling B, false. What about others? Maunia, Anaka, and Vidushi is also going with B. What about Maunia and Anaka? Now they will also go with B. Any other options? Maunia and Anaka, B. Yes, very good. Because chemical synthesis of steroids are more complex than microbial transformation. That's why we are going for microbial transformation, because microbial transformations are less complex. We can go for dehydrogenation, hydroxylation, whatever. Okay, very good. And the third and the, I think the one more question only. Yeah, the third and the final question goes to Anaka. Dash is an antiviral human protein. Options: interferon, interleukin, HGH, TNF. Anaka, Dash is an antiviral human protein. Question goes to Anaka. Your option: A, B, C, or D. Anaka, Dash is an antiviral human protein. Antiviral. I have used the same term while I was taking class. Any answer from Anaka? Dash is yes, B, interleukin. Very good. What about others? What about others? Maunia, Vidushi, and Shalini. Dash is an antiviral human protein. Interferon, interleukin, HGH, TNF. Anaka has given the option B. Shalini has also given the option B. What about other two? Now everybody will go for B. What about other two? Vidushi and yes, Vidushi, very good. B. Once more. Yeah, all of you have gone for B. That is interleukin, but I think the answer is A, interferon. Let me go back. Let me go back. Dash is an antiviral human protein. See, I have explained it here. Interferon is important in the defense against viruses. So interferon is the antiviral human protein. So the mark goes to the quiz master. Is it clear? All of you? Yes. Okay, our organizers will be sharing these slides with you. Please go through this chart which I have taken from this. These charts are very important, you know. All informations are there in the chart. Since it is applied biology, they will be asking like this. I hope so. We cannot tell about their exam pattern. Okay, with this, our today's topic comes to an end. Over to the organizers.
Okay, thank you, sir. So, students, if you have any question regarding to the topic, please comment. Or if you understand today's topic, please comment yes in the comment section. That let us know.
Again, let me repeat. If you have any doubt while you are learning or something, either you can ask in the class or please use my mail ID or even WhatsApp number. Fine. It's always available. It's always online.
Yeah, thank you, sir. Thank you. It was really very informative. And so, if you have no doubts, then one by one you can leave the meeting. Thank you. That's all for today.